Freezing African elephant semen as a new population management tool. As the genetic diversity and availability of captive elephant breeding is on the decline, it is important that a new means of genetic material is imported to improve the sustainability of the breeding program. The solution? Importing the semen selection from wild bulls via sire preservation. The less and more secluded of the wild populations are at higher risk of inbreeding, with the ramifications being a reduction in the fitness of the population, which is also true in the case of captive populations. This study investigates the means by which diversity of the population's gene pool can be sufficiently extended by importing from the wild. For this study, 14 African savanna elephant bulls were selected from the Finda Private Game Reserve in the Republic of South Africa and were aged between 14 and 36. The animals were anaesthetised for the extraction process by the means of gun. Semen was collected by a method called electroejaculation. The probe was inserted preceding an ultrasound, which was used to identify the location of relevant organs. The probe was injected and the electrical coupling was executed alongside a manual massaging of the pelvic and penile sections of the urethra to extract the sperm. The sperm was amassed in a collection bag that was placed over the penis. Cryopreservation. After diluting the sperm samples with freezing extenders, the semen was frozen using a directional freezing machine. The frozen samples were then stored in liquid nitrogen. As can be discerned from Table S1, the mobility of all the samples has no significant difference between them. Figure 1 compares the treatments of glycerol percentage and exposes that when the semen is placed in the 7% glycerol, this establishes the best mobility outcome. Only when both years' results were combined were the results rendered significant for acrosome integrity and morphology, as can be seen in Table 3 and Table 4. There are 4 major obstacles that have to be addressed in this study. Obstacle of protocol that hasn't yet been established for many species in regards to sire preservation. The logistics of successfully creating a program that would occur frequently and would be extremely expensive and require a large amount of funding. Permits for certain tests and liabilities have to be obtained and considered. And finally, the actual artificial insemination process, having a deep enough understanding of all the parts and techniques involved to create a successful outcome. Another liability to be aware of is the health circumstances of the wild animals, as monitoring their health and disease status is extremely difficult to maintain. And as a consequence to this, there is a large amount of problems for the continuity of the program that could arise. And the conclusion? It can be done. Furthermore, if the samples of the semen are centrifuged and the use of directional freezing for sire preservation occurs after the samples are diluted to 7% glycerol in the extender, the outcome is likely to be successful. As proof to this, there has recently been reports of the first pregnancy in elephants via this method in captivity. Thank you.